Good morning, students. Our today's topic is Human Genome Project. As we know, genetic makeup of an organism or an individual lies in the DNA sequence. If two individuals differ, then their DNA sequence should also be different, at least at some places. These assumptions led to the quest of finding out the complete DNA sequence of human genome. Genome, what is genome? The haploid set of chromosome is known as genome. Haploid means n number of chromosomes. A very ambitious project of sequencing human genome was launched in the year 1990. Human Genome Project was called a mega project. Now the question can be asked, why Human Genome Project was called a mega project? Answer is, first point, human genome is said to have approximately 3 into 10 raised power 9 base pairs, and if the cost of sequencing required $3 per base pair, the total estimated cost of the project would be approximately 9 billion US dollars. So this much huge amount is required for sequencing the base pairs. Further, if the obtained sequence were to be stored in typed books and each page of the book contained 1000 letters and each book contained 1000 pages, then 3300 such books would be required to store the information of DNA sequence from a single human cell. So this much information, these many books are required to store the information. The enormous amount of data expected to be generated also necessitated the use of high-speed computational devices for data storage and retrieval and analysis. For this, highly specified computers are required to store all this information. Human Genome Project was closely associated with the rapid development of a new era in biology called bioinformatics. In bioinformatics, the DNA sequences is stored using computers. So with the use of computers, the information of DNA is stored. What are the goals of Human Genome Project? First, identifying all the approximately 20,000 to 25,000 genes in human DNA. In the human DNA, 20,000 to 25,000 genes are present, and to identify the genes, this is the goal of HGP. Second, determining the sequence of the 3 billion chemical base pairs that make up human DNA. Next, storing this information in databases in computers, then improve tools for data analysis, same point, transfer related technologies to other sectors such as industries, then sharing the information of base pairs with other industries like computer industry, etc. So when we are doing all this, many ethical, legal and social issues arises. To solve the issues related to this project was also the goal of HGP. The Human Genome Project was a 13-year project coordinated by the US Department of Energy and the National Institute of Health. These departments help in Human Genome Project. The project was completed in 2003. Knowledge about the effects of DNA variations among individuals can lead to revolutionary new ways to diagnose, treat, and someday prevent the thousands of disorders that affect human beings. If we know the sequence of DNA, we can cure many diseases. Many non-human models such as bacteria, yeast, nematode, Drosophila, rice, etc. have also been sequenced, their DNA sequence. We also know their DNA sequence. Now, what are the methodologies What are the methods that are used for Human Genome Project? Mainly two methods are used. First one is expressed sequence tags. Second is sequence annotation. These are the two methods. So first, express sequence tag. In this, all the genes that are expressed as DNA as RNA are referred to as expressed sequence tags. The DNA changes into RNA. So all the changes, all the genes which appear in RNA, mainly exons, because introns are removed. So in this method, only exons are considered which are present on the RNA. The other took the blind approach of simply sequencing the whole set of genome that contained all the coding and non-coding sequences, and later assigning different digits in the sequence with functions. This is known as sequence annotation. Annotation means explanation. In this, the whole DNA was explained without considering whether it will represent in the RNA or it will not come. So whole DNA sequence is represented in sequence annotation. Then afterwards their functions were assigned. Here in this, exon, intron, everything are present. For sequencing, the total DNA from a cell is isolated and converted into random fragments or relatively smaller sizes and cloned in a suitable host using specialized vector. For this sequencing purpose, first we have to take the DNA. DNA is taken from the cell and that is cloned. Clone means many copies are made of that in a suitable host using special vectors, and then from that clone they study the sequence of the DNA. The cloning resulted into amplification of each piece of DNA fragments so that it subsequently could be sequenced with ease. When we clone that DNA, that produces many copies, and then it becomes easy to study that DNA fragment. In this, mainly the host used is bacteria and yeast. In that, the host mainly used are bacteria and yeast, and if bacteria is used, then the vector is BAC, that is bacterial artificial chromosome, and when yeast is used, the vector is yeast artificial chromosome, that is the artificially prepared chromosome that is used as a vector. Learn the names BAC and YAC. Question can be asked. Write the full form of BAC and YAC and what is their function. The fragments were sequenced using automated DNA sequencers that worked on the principle of method developed by Frederick Sanger. So only just learn one line, that these segments are then sequenced using automatic devices or computers. This was developed by Frederick Sanger. The Frederick Sanger again gave one more contribution, that he determined the amino acid sequence in proteins. He explained about the amino acid sequence in proteins. So based on that, he gave certain sequencing in the DNA also. These sequences were then arranged based on some overlapping regions present in them. What are overlapping regions? Overlapping regions are a pair of adjacent genes whose coding regions are partially overlapping. Some part in the genes overlap each other, that is known as overlapping regions. This required generation of overlapping fragments for sequencing. Alignment of these sequences was humanly not possible. Therefore, specialized computer-based programs were developed. For this sequencing, special computers were used and they gave the complete sequence. These sequences were subsequently annotated, explained, and were assigned to each chromosome. After they were used by the use of computers, the DNA sequence were explained. The sequence of chromosome was 1 was completed in May 2006, and this was the last of the 24 human chromosomes. Why 24 chromosomes? 23 pairs, and because 22 are autosomes plus sex chromosomes X and Y, both are different in structure, so total 24 chromosomes were sequenced. But chromosome number 1 was the last to be completed in 2006. Another challenging task was assigning the genetic and physical maps of the genome. So, this paragraph we will study in DNA fingerprinting. When I will explain DNA fingerprinting, then only you will understand polymorphism, restriction, endonuclease, recognition sites, all these terms. So at present, just read till here only. Important questions from him. This is, what is Human Genome Project? Why it is called Mega Project? What are the goals of Human Genome Project? What are the methodology used? So these are the important questions. If you have any doubt, you can ask me. Thank you.